next part of the physiology section of the male reproductive system is spermatogenesis, which literally means forming sperm, which are the male gametes. And there's the definition right there. So we already know it occurs in the seminiferous tubules of the testes. It begins at puberty, which is around 14 years of age. The adult male makes 90 million sperm cells daily, and spermatogenesis takes 64 to 72 days from the primary spermatocyte to an immature sperm. So we need to go through this process. And of course, from an immature sperm, it takes about 20 to 24 days to become mature. So let's look at this process in a little more detail. So here are the seminiferous tubules, which we've seen already, that are located in the testes and where the sperm cells are produced. The seminiferous tubules are thick, stratified epithelium, which we can see right here. And here we're looking at the lumen, the inside of the seminiferous tubule. So there's the wall around the lumen. There's 4 important cell types, which we'll see. So we're just looking at a little cross-section of a seminiferous tubule right here. And we have these sustenocytes. Think of sustaining, sustenocytes. They surround, they support, and they nourish the developing sperm. And they're pointing to sustenocytes in 2 different seminiferous tubules, because there's the basal lamina or wall that's dividing to 2 seminiferous tubules. Here we have arrows pointing to all these other cells in the wall of the seminiferous tubule. Those are spermatogenic cells. They're in the tubule epithelium. They're sperm-forming cells, as their name implies, spermatogenic cells. And they're surrounded by sustenocytes, which is the first type of cell that we went over. Then we have interstitial endocrine cells. And if we look to where they're pointing, they are not inside a seminiferous tubules. They're actually located between seminiferous tubules, which is why they're called interstitial endocrine cells. And think endocrine, well, they must produce a hormone if they're endocrine cells. So they're in the connective tissue between tubules, and what they secrete is testosterone. All right, so sperm is produced in the seminiferous tubules, but the hormone testosterone is secreted from the interstitial endocrine cells. Then we have myoid cells. Myo means muscle, oid means resembling, so muscle-resembling cells, myoid cells, which they're pointing to some here and here. They're smooth muscle-like cells, right? Resembling muscles, myoid, smooth muscle-like cells, that they surround the seminiferous tubules, and they may have a contraction-like ability to help propel material along inside the lumen of the seminiferous tubules. So those are the important cell types, but let's look at the process of spermatogenesis in some more detail. So here we're looking at the wall of a seminiferous tubule. This would go all the way around the lumen. There's the lumen inside a seminiferous tubule. So if you look back here, we can see, there we go. Yeah, we can see there's the lumen, there's the seminiferous tubule itself. So in that picture, what I'm going to, we're just looking at a section where we see the seminiferous tubule and some of the lumen. So there's the basement membrane or basal lamina right there. So if we just take a section of that, that's what we're looking at here. So we have this basal compartment against the basal lamina here, and then we have this adluminal compartment, which we'll become familiar with, and there's the lumen of the seminiferous tubule. So the process starts at the basal lamina and ends with these immature sperm cells in the lumen. So when we look through this process, we can see it inside the tubule here, seminiferous tubule, or just look at the cells in general. This is the process of spermatogenesis. And it begins with a stem cell, which is called a spermatogonium, which is diploid 2N, which goes through mitosis. So basically, from one cell, we get two cells. We know mitosis from chapter 3 in A&P Part 1. So now we have two of these diploid cells. One is going to stay put. So the type A daughter cell is going to stay at the basal lamina, so it can do the same thing all over again. Where the other copy is actually going to go through spermatogenesis through this process. So this type B daughter cell will now enter spermatogenesis and go through meiosis, which we talked about at the beginning of this chapter. So here's our diploid cell, which becomes a primary spermatocyte. When it goes through meiosis 1, we get diploid to haploid. So we get these two cells, which are now secondary spermatocytes. So a primary spermatocyte is diploid, and after meiosis 1, the secondary spermatocytes are now haploid cells, and they're actually connected. So that cytokinesis isn't quite complete, and they have this bridge formed between them. Let's go through meiosis 2, right? So that divides the sister chromatids, which we learned at the first part of this chapter, and there's our 4 haploid cells, and that's basically meiosis, which you can review at the beginning of this chapter, from diploid, one diploid to 4 haploid cells. So we call these 4, which are all joined by these little bridges, right? They're all connected in spermatogenesis. We call these spermatids. And this is early spermatid, which just look like normal haploid cells. Late spermatids now are starting to look like sperm cells at this point. And to get from a spermatid to an immature sperm cell, this process, which we'll need to go through, is called spermiogenesis. Don't confuse that with spermatogenesis. Spermatogenesis is this whole process from these stem cells to immature sperm cells, but spermiogenesis just takes a spermatid and turns it into an immature sperm cell. So careful with some of this terminology. A lot of terms sound similar to each other, like spermatogonium, spermatocytes, primary and secondary, spermatids. A lot of confusing or easily confused terms. And we can see how, as this process occurs, it moves through the seminiferous tubule, the spermatogonium, right? We get the primary spermatocyte, which divides into haploid secondary spermatocytes, which goes through meiosis 2 to get the spermatids, and then spermiogenesis ends in the lumen, in the seminiferous tubule. We'll see these tight junctions again, come back to it and why they're there soon. So here's just spermiogenesis, which takes approximately 24 days. That's transformation, which we saw on the previous slide, transforming a spermatid into a functional sperm. So there's our spermatid. There's the nucleus, centrioles. Here we see an acrosomal vesicle, which we'll get into more a little later. There's the Golgi apparatus. And what happens is that this sperm or spermatid will begin developing a flagellum, a tail, which its job is to propel the cell, unlike cilia, which propels material outside the cell. And then we can see the cytoplasm beginning to disappear by the time we get to the functional sperm. So the head will contain the nucleus and something called an acrosome, which we'll describe on the next slide, where the tail would be the flagellum and the excess cytoplasm will get discarded. So that's the head. We have a midpiece, which connects the head to the tail, which is the flagellum. So let's make that official. In spermatogenesis, summary of events in the seminiferous tubules, we have major regions of the sperm, the head, the genetic region, because there's the nucleus, and it has a helmet, or a helmet-like acrosome, and that acrosome is important. Remember the acrosome right there on the tip? That acrosome is important because get a better pointer, there we go. It contains hydrolytic enzymes, and without that, that sperm cannot penetrate the egg in the female. So that acrosome is there to break through the all the supporting material surrounding the egg, so the sperm can actually penetrate the ova in the female. So the midpiece of the sperm is a metabolic region. That's where the mitochondria is. So that's where the ATP is produced. Of course, we need ATP to move the flagellum. And the tail is the locomotor region, which includes the flagellum for motility.